Welcome to Botany class. Chapter 3 Enzymes. Part 3 Classification and Nomenclature of Enzymes and Cofactors. In this we will discuss about enzymes have been discovered, isolated and studied. Most of these enzymes have been classified into different based on the type of reactions they catalyze. Enzymes are divided into 6 classes with 4 to 13 subclasses and designated accordingly by a 4-digit number. This is very important in a NEET. Other topic, first one, oxidoreductases, dehydrogenases. Enzymes which catalyze oxidoreduction between two substrate S and S product. Example, S reduced and S1 oxidized is gives to S oxidized and S1 reduced. Example, malate plus NAD plus malate dehydrogenase enzyme activate on that and it gives rise to oxaloacetate plus NADH plus H plus. Second one, transferases. Enzymes catalyzing a transfer of a group G other than hydrogen between a pair of substrate S and S prime. Example, S G group plus S prime is gives rise to S prime and G. Example, glucose plus ATP action of exokinase is gives rise to glucose 6-phosphate and ADP. Here on glucose 6 carbons, 6th carbon which attaches phosphate group. That's why we called as glucose 6-phosphate. Hydrolysis, third type of enzyme which is catalyzes hydrolysis of a ester, ether, peptide, glycosidic and double C with a single bond, either carbon with halide or product with a nitrogen bonds. Example, fructose 1,6 bispage plus H2O gives rise to fructose 6-phosphate plus inorganic phosphate. Here the enzyme which is hydrolysis fructose 1,6 bisphosphatase. So when we called fructose 1,6 phosphate, because of fructose have a 6 carbons in that number 1 6 and 6 carbon which contain phosphate groups. That's why we called as a bisphosphate. It indicates that 2 phosphates are present. Lyases, enzymes that catalyzes removal of a group from substrates by mechanisms other than hydrolyzing, leaving double bonds. Here if you observe, carbon one group which is attaches with X and one carbon attaches with Y gives rise to XY and a C double bond C. Here the products are only the carbon group. Example, arginosuccinic acid gives rise to arginine plus fumaric acid by the action of arginosuccinase enzyme. Isomerases includes all enzymes catalyzing interconversion of optical, geometric or positional isomers. Examples, ribose 5-phosphate gives rise to ribulose 5-phosphate. Here action of enzyme ribose 5-phosphoisomerase. The answer, the phosphate which is attaches with the 5th carbon, due to that we write 5-phosphate. Ligases, 6th one, enzymes catalyzing the linking together of a 2 compounds. Example, enzymes which catalyzes joining carbon oxygen, carbon sulfur, carbon nitrogen, phosphorus oxygen bonds. Glutamic acid plus NH3 plus ATP gives rise to glutamine plus ADP plus IP. Here the enzyme which is catalyzes glutamine synthetase. Here glutamic acid which convert into adding of amino group, glutamine. Above classification provides for a 4-digit code to identify individual enzymes. For instance, glucose 6 phosphotransphorases has the enzyme code. The first digit of the code indicates the major class of the enzyme, while the second and the third digits indicate subclass and the subclass respectively. The last digit of code is the serial number of an enzyme in a particular subclass. So this is a NEET bit which you can remember 1, 2, 3, 4 sequences, and the question may be asked how many digits are there in the enzyme coding. Then cofactors. In this, enzymes are composed of one or several polypeptide chains. There are number of cases in which non-protein constituents called cofactors are bound to the enzyme to make enzyme catalytically active. There are 3 kinds of cofactors may be identified. One is prosthetic groups, second coenzymes, and third metal ions. Prosthetic groups are organic compounds that are distinguished from other cofactors in that they are tightly bound to the apoenzyme. Example, peroxidase catalase. Coenzyme are also organic compound, but their association with the apoenzyme is only transit, usually occur during the course of catalysis. Coenzyme serves as a cofactor in a number of different enzyme catalyzed reactions. The essential chemical component of many coenzymes are vitamins, either NAD or NADP, vitamin niacin. And third one, a number of enzymes required metal ions for their activity which form coordination bonds with the side chains at active site, and at the same time from one or more conditioned bonds, coordination bonds with the substrate. Example, zinc is a factor for the proteolytic enzyme carboxypeptidase. So lastly, catalytic activity is lost when the cofactor is removed from the enzyme, which testify that cofactors play a crucial role in the catalytic activity of an enzyme.